Hello, everyone. Thanks for joining us. My name is Martin Westgate. I'm Science Advisor to the Atlas of Living Australia. And thank you for joining us for this webinar. This is our second of two to celebrate 10 years of operation as an organisation. For those who don't know, ALA is a data repository. We store and share information on Australia's biodiversity, but we don't do a lot of research in-house, despite it being our role to support research. And so we thought a nice thing to do for our 10-year celebration would be to hear from people who do use that data and data from other places and are really on the cutting edge of understanding where what's going on with biodiversity in this country. And so I'm very thrilled to be able to share three of those speakers with you today. So we've got Melodie McGeoch, who's coming from La Trobe University, August Hao from School of Biosciences at the University of Melbourne, and Jane Elith, who's also at the University of Melbourne, professor there, and a former Life Scientist of the Year. So we've got a wonderful panel. I'm not going to take up too much time. I would like to acknowledge that while everyone that we're speaking to today is down south in Victoria, I'm calling today from Canberra, so we're on Ngunnawal country. And I'd like to pay my respects to the elders of the Ngunnawal people, past, present, and emerging. On a much less interesting and more mundane note, you're probably viewing this through a particular browser. You should be able to see some, on the right-hand side of your screen, there is a section for questions and a chat function. Feel free to drop some stuff in there. Tell us where you're listening from. We'd love to hear from you. And we will have time for discussion at the end. So if you want to put any questions in there, we'd love to hear those. There's also some polls in there if you want to tell us some information about where you heard about this webinar. We'd love to hear your thoughts. So I don't want to take up our time because the other three speakers are much more interesting than me. So I'd like to pass over to Melodie first. Melodie, if you'd like to share your screen, we'd love to hear what you have to say. Thanks.
Thank you, Martin. Can you hear me? It's all good. Excellent. Thank you for that lovely introduction. And yes, this is really a wonderful way to celebrate 10 years of the Atlas of Living Australia. So I'm thrilled to be talking with you all today. I thought that we'd spend the next 12 minutes or so thinking and talking about common species and making the case for the fact that common species are comparatively neglected, both in terms of biodiversity theory, but also, of course, in conservation efforts. We tend to pay a lot of attention to particular species, and for a range of reasons. We have iconic species, much loved species that have very strong cultural significance. And some of these species also often have the ability to draw significant finances for conservation. We also pay attention to rare and threatened species because we consider that they have existence value, and we therefore invest fairly significantly often in efforts to prevent their extinction. Now, traditionally in conservation, there's a very long history, of course, of two approaches to conservation: the species-based approach and the habitat approach. And the long-held wisdom is that by focusing specific attention on particular species that are threatened with extinction, along with a focus of attention on protecting habitats, protecting landscapes, we should be able to conserve both rare and common species. However, over the last few decades, we've started to realize that that assumption is flawed, because there are now many species that we know that just a few decades ago we would have thought of as very common species, and that these species are now in precipitous decline. Just to give you a couple of examples, the house sparrow across the US and across Europe has undergone between 70 to 80% decline in abundance since the late 1960s. And the white rhinoceros in Africa has lost 99% of its population, and there's been a particular sharp decline in numbers over the last decade as a consequence of a dramatic increase in levels of poaching. So we know that the conservation status of species can decline very rapidly, And we know that this decline is quite noticeable in those common species that we do tend to pay attention to for one reason or another. But perhaps before we take the story any further, we need to think a little bit about what we mean by common. We're quite used to defining rare species and quantifying forms of rarity thanks to the IUCN red listing criteria for threatened species. But when it comes to defining common species, we can in fact use the same dimensions or variables in which to quantify them. And this includes some measure of the range of a species, either its extent of occurrence or area of occupancy, but also some measure of local population size or abundance. And then we're interested in whether the trends in a particular species are increasing, declining, or comparatively stable. And in the same way we think about rarity, we could think about this cube as being all common species except for the purple block in the lower left, which constitutes species with small ranges and low average population sizes, for example. This means that all the other species in this cube have properties that mean that they are in some way common. If we just take one of these properties, range or species distribution, and we have a look at a frequency distribution of the numbers of sites occupied by a particular species. The shape of this frequency distribution is fairly well known, and it commonly looks like this. And what we've paid a lot of attention to is the fact that most species in the world have narrow ranges or are rare, at least according to this dimension. However, if you define a common species as anything that is not rare, then it means that the cumulative total of species in all these bars on the right could be considered species that are in some ways common. So that's just in terms of the geographic range of rare or common species. What about species abundance? So in this figure here, we're looking at the frequency of abundance, of species abundance, but in a slightly different way. So what we are focusing on here are birds, because we have good data for birds. And on the y-axis, we have cumulative abundance. So every point here is a different bird species, and these are data on bird species across the world that are currently recorded as having declining trends. So there's about 2,100 odd species in this figure. Each point is a species, and we're going from the most abundant species to the least abundant species and accumulating their total estimated population sizes. And each species is color-coded according to whether it's least concern on the IUCN Red List or, for example, threatened on the IUCN Red List. So what you can see in this figure is that the most abundant species are, not surprisingly, all classified as least concern, even though they do have declining population sizes. But what is also quite remarkable about this figure is that only 164 species make up 95% of the total abundance of birds across these 2,000 odd species. So from a conservation status perspective, we might not be too concerned about the decline in conservation status from a species conservation perspective of these species that are in green and currently considered to be least concern. But it certainly seems as though if they're declining, we're losing a large proportion of bird biomass on the planet as a result. So what is the cost then of not protecting our common species? In this figure, we're looking at the thresholds of declines in population size of species that trigger a change in IUCN Red List status. So what does it take for a species that is currently least concern to be reclassified as near threatened or vulnerable? Well, according to the IUCN red listing process, it takes a decline of a 20% population size over a time frame of 10 years or 3 generations. So let's take our willow warbler that we saw in the previous slide, was the second most abundant species undergoing declining trend or undergoing a population declines. So there are an estimated 500 million willow warblers globally. If we take the body mass of an individual willow warbler and we say that a 20% decline, we multiply that up by an individual willow warbler body mass, we get about 10,000 tons of lost biomass in willow warblers to trigger a change in conservation status. Now, I don't know whether you prefer your currency in terms of gold bars or in blue whales, but a 10,000 ton loss of willow warbler equates to about 68 blue whales or about 750,000 bars of gold. So while we might not be worried about the conservation status of the willow warbler or its threat status, we certainly need to be thinking about the functional consequences of the loss of biomass in common species. Now, if we think about willow warblers declining, and let's move to an Australian context here, and we're looking at bird species across southeastern Australia. And if we think about the fact that we're losing individual birds from populations across landscapes, we know, of course, that these losses are not spatially homogeneous. And that assumption or that statement is supported by the data here. So what we've got is 45 species that occur across these three regions in southeastern Australia, these three biogeographic regions, the Eastern Mallee, the East Coast, and the Southeast Mainland. And the percentages in the intersections are the percentages of species that have the same estimated population trends across all three regions. So you can see that only 49, overall only 33% of species have the same trend across these regions. So that means about 2/3 of the species overall are actually either declining or increasing or stable, but basically their population trends vary spatially. So if we're interested in the sustainability of ecosystems, the sustainability of landscapes, the maintenance of biomass in landscapes, we really do need fairly local scale data on species population sizes and abundance, including species in that qualify, or particularly species that qualify as common. All right, well, if that is the case, how much do we already know about whether common species are declining or not? Well, this is a distribution of survey efforts of bird species across Australia, and using a combination of simulation and comparison with data observation, so simulating survey design, including detection efforts and so on. I refer you to the paper at the bottom for more detail. These statistics here show the percentage of common species present in each of these localities for which we can be confident that there are enough power in existing bird data surveys to detect a 30% decline in population size. So you can see that in Darwin, there's a very low percentage of species for which we have data with enough power to detect fairly dramatic population declines, 30%, compared to, for example, Melbourne, Melbourne and Sydney, that are sort of comparatively better in terms of the adequacy of our data to detect population declines in common species. I want to end with just an example of a common species that's currently undergoing very dramatic decline. This is a plant species on Australia's Macquarie Island in the subantarctic, and it's called the Macquarie Island cushion. It is a foundation species widespread across the island. The genus is widespread across the subantarctic, and over the last decade or so, the species has undergone an average of around about a 30% loss of cover across the entire island. However, if you look at the data, there's been much more dramatic decline over the north of the island than the south of the island, and there are in fact some landscapes in the north that are now completely denuded of all vegetation because they've lost this foundation species. And this has happened very rapidly in response to warming and wetting on the island, and the increase in the prevalence of a soil pathogen as a consequence that negatively affects this plant. So in the bottom here, you can see this browning, which eventually just blows away and leaving sort of denuded gravel landscapes. So to conclude, if we think about the fact that species threat status actually motivates the collection and collation of population data, it's not surprising perhaps to look at this figure over here, which shows the three important dimensions of data and information that we need for species populations: area of occupancy, extent of occurrence, and population size, color-coded by whether they're critically endangered to least concern. And you can see for all taxa, there's a decline in the percentage of species that have actually been assessed as their evaluated conservation status improves. So this just basically means that there are far fewer least concerned species assessed per taxon than there are critically endangered species, with the exception of birds. So this is a bit of a downward spiral of danger if we just keep measuring only those things that are critically endangered. We really do need to balance our focus and start better measuring the population sizes and distributions of common species. So I have a lot of collaborators to thank, particularly David Baker and Kath Dixon, whose work I drew on quite strongly here. But I'd very rapidly like to just end off by congratulating the Atlas of Living Australia on their 10-year anniversary. If protected areas are considered to be the cornerstone of biodiversity conservation, then the Atlas of Living Australia could be thought of as the cornerstone of biodiversity knowledge for Australia, and is absolutely essential for preserving our natural heritage into the future. So congratulations to the last 10 years of innovative and dedicated people who are the Atlas of Living Australia. Thank you.
Lovely, thanks for that, Melody. That was a really fascinating talk. And I've got a list of questions that I'll be coming back to you with at the end, but we'll save those for when everyone has spoken as well. So our next speaker is August Howes from School of Biosciences at the University of Melbourne. August, would you mind sharing your slides next? Is that all right?
Sure.
Lovely, thank you for that. I'll hand over to you.
Thank you. Can you see me all right? Yep, perfect. All right. Thanks, Martin, and thanks for coming to my talk, everyone. My name is August. I am a PhD candidate at the University of Melbourne. And today I'll be presenting a chapter of my PhD thesis and my work on the biogeography of Australian fungi. So, I would like to first start by acknowledging the traditional owners of the land across the continent of Australia, whose ecosystems I study, and in particular, the Wurundjeri people of Kulin Nation, upon whose land I live and work. I want to pay my respects to the elders past, present, and emerging. So you see that in my title, I pose this question: can open access biodiversity data help us study the biogeography of Australian fungi? I want to break this question down and explain why I think it's worthwhile asking by first starting by explaining its three key concepts. Open access biodiversity data, which I'm sure many of you are very familiar with, are the type of data obtainable from the Atlas of Living Australia and from other repositories such as GBIF and iNaturalist. In the context of studying biogeography, these data are valuable because they provide information on the whereabouts of species occurrences, such as illustrated on this map here, where the dots represent records of species occurrences. Open access data are really a vast and growing resource of information, particularly for taxa that have been historically understudied and undercollected, such as fungi. There are now more than 2 million records of fungi at LA alone, which is really a tremendous amount comparing to what it used to be. And secondly, I want to explain why biogeography is worthwhile studying, especially for fungi. So biogeography is the study of organisms in space, and it constitutes a foundational knowledge upon which we can better understand nature and to better conserve the natural biodiversity. And evidently, to know something or to study the location of species, you need information or data on where they are found. And this is where open access data can really help. Unfortunately, biogeography of some taxonomic groups are really little known, typically because they're hard to collect data for. This can be, for example, hard to find species such as fungi or bacteria. So lastly, I want to touch on why Australian fungi tie all these different concepts together. Fungi are an entire kingdom of life that are really important in terms of the ecosystem functions they play, and also because they are vastly diverse. So there are more than 20,000 species on LA alone belonging to the kingdom of fungi, which is also expected to be only a small proportion of the total undescribed biodiversity. Many of you might be familiar with the typical types of fungi we see, which are macrofungi, meaning that they produce visible fruiting bodies, usually in the shape of mushrooms seen here. But the kingdom of fungi also includes life forms such as lichen and many different forms of microfungi, which are too small to see with the naked eye. And because many fungi are so hard to see, they're actually a little bit hard to study, especially in terms of collecting locational data. So our understanding of fungi is really minimal compared to plants and animals in Australia. And this has led to the consequence of poor fungal conservation, simply because we don't know enough about where they are. So to be able to fill this knowledge gap, open access data are a very important resource for fungi because they represent the vast majority of data that's available out there. And with 2 million records in LA alone, I think this is a great opportunity through which we can learn more about fungi and do a better job at conserving them. However, these data also come with their challenges. For example, if we just look at all the fungal records on the LA for the continent of Australia, we get this map, and immediately you see features that might give you a problem if you're using analysis. As example could be all the records that's falling into the ocean, and all the gaps in the outback where there aren't any recording. I think the challenges of open access data can be summarized into two major categories or aspects. The first relate to quality issues, meaning that there are errors in the geocoordinates or taxonomic names of open access records. Some of these can be obvious, such as this clump in the ocean here on the map, but often they can also be hard to find when they are clustered together with correct records. And because of these errors, it is important that we implement some sort of quality tests that allows us to filter the data and to check for errors as much as we can. The Atlas of Living Australia actually has a really nuanced approach to how they do this, which is also described in this paper here. But what we found is that it's also worthwhile for end users of data to run their own additional tests that benefit their own use cases the best. The second aspect of challenges are what I would call incomplete coverage. So this is less of an error and more of a feature of the data that we need to work with. And coverage are incomplete often because they're areas that are simply difficult to visit by people, and therefore we have very little recording over those locations. This could be problematic because it can introduce bias in the data by confusing information about species occurrence intensity with recording intensity. In other words, we simply do not know whether a place is really unoccupied by species or if it's just not visited by someone. Obviously, the best way to improve this is to increase sampling effort in the future, particularly in locations where there hasn't been any samples or records so far. So, having explained those challenges, now we see that this question is not really straightforward to answer, and we need in-depth exploration of open access data for Australian fungi, and particularly we need to sort out the quality issues they may possess and to explore the data in more detail to be able to see what sort of information we can unpack from them. So for the next set of slides, I just want to walk you through what we did with Australian data and to present some preliminary results on what we can get out of it. So the first thing we need to address are the quality issues, which we tackled through a reproducible pipeline of different quality tests. And don't worry about the details on this slide. I'll walk you through the main framework we've taken. So in essence, we downloaded more than 1 million records of open access fungal data from 5 major repositories hosting fungal data. And we collected these data sets and subjected them to an iterative and reproducible pipeline consisting of different steps, meaning to target different aspects of quality issues. And what I want you to focus on is that at the end of the pipeline, we ended up with only a quarter of the initial data. Although many of the discarded data are simply discarded because they're not at species level or because they're duplicates, what we found is that the cleaning process really quite substantially changed the content of the data set. But I just want to emphasize again that many of the data discarded here are not erroneous. They're simply not what we were looking for in the final data set. So having addressed the quality issue to the best of our ability, the next thing we want to do with the data set is to explore its coverage in terms of different dimensions. Here, we're aiming to uncover diagnostic information about what the data may reveal and what the data might be lacking if we use it for different types of geographic analyses. And I'll be presenting the results in terms of the data breakdown over these dimensions. Could I please ask you not to share these figures here because they're part of the manuscript that we're yet to publish. So going over the main results, in terms of space, what we are looking at are spatial coverage of fungal data from the clean data set when they are aggregated to 100 by 100 kilometer grid cells. So looking at this map, the first thing we really immediately see are all the gray cells representing places where there aren't any recordings. So there's quite a lot of them in the outback, but also in the tropical north. We also see cells with dark purple colors. These are actually areas where there's only one record, so it's not much better than having no data. And this is really in stark contrast with well-recorded areas. So you might see the most lit up areas in Melbourne and in Hobart, where there are roughly about 20,000 records in each of those cells. Although the spatial coverage is patchy and incomplete, environmental coverage shows a different story. This is a little bit complicated, so I'll walk you through the details. What we're looking at here is a multivariate environmental similarity surface. This is a tool we use to show which location across Australia are environmentally outside the range of existing data in terms of the environmental variables we use here, which relate to climatic and soil factors that we believe are relevant to fungi. You can read about the methodological details in this paper here, but in essence, what I want you to focus on are these purple bits scattered across the outback and certain parts of Australia. These are the only locations that are outside the environmental range of existing data in terms of the environmental variables we considered. So this is really quite optimistic, meaning that comparing to this patchy spatial coverage, we have more complete coverage in the environmental space, meaning that some of the locations where there haven't been any recording so far are at least environmentally similar to some other locations that have been recorded. This is quite useful for any sort of species environmental modeling work, which I believe Jane will talk about in a bit. We next move on to the temporal trend in the data set. So the data really covers a large range of temporal domains, starting all the way from Captain Cook in the 18th century. But we see that data collection has really taken off in the recent decades, especially since the 2000s, and especially with the increase of human observations. So this corresponds to the rise of citizen science, and especially the ability to record photographs of species with mobile phone, which has really helped a lot to bulk up the number of records in recent years. Now we're moving on to species accumulation. So what we're looking at here are two species accumulation curves. And we're not going to the method details, but what I want to draw your attention to is that these represent how many new species are being discovered as we increase sampling effort, either over space, so that's on the left, or over time, so this is on the right. This is specifically for the block that for the 100 kilometer block over Melbourne, which is the best collected area. And this is over as we increase the number of blocks across Australia. So on both graphs, what we see is that the curve does not trend towards equilibrium or asymptote. So as we increase our sampling effort over either space or over time, we discover new species, even in areas where there have already been a lot of records. So this really goes to show that if we increase our sampling effort, or if we go out and collect more data, we'll likely end up discovering new species across the whole continent. So now I want to present some results about the collection effort and the collector themselves. So what we found, consistent with many other open access datasets in other countries, is that some prolific super collectors contribute disproportionately large to the dataset. In fact, the top 10 collectors contribute to almost 50% of all data, which is really a sizable amount. So here are two figures of the collection footprint, if you will, of two particularly prolific collectors. And not sure if you can see very well, but we can often see the home location of collectors. So this one being in Canberra and this one being in Melbourne, where they collect most often. But we also see that they're happy and willing to travel far and wide and to cover a wide variety of locations to collect data from. But we also see evidence that their collection footprint are being restricted by roads or being proximity to major city. This is also reflected further if we look at how collection locations coincide with infrastructure or accessibility to cities. So on the map on the left, what we have in the background are travel time to the nearest city in terms of hours, and the black dots represent locations of collection in my clean dataset. We see a strong pattern that almost all collection locations are either close to major cities or they coincide with roads. So it really goes to show that collection effort appears to be driven by accessibility, and places that are close to city are collected more often. The same pattern can be seen if we plot the frequency of travel time to cities among places with collection. So the bars represent areas of places where a collection has happened, and its distribution is vastly different from the black line here representing the Australian average. So this further goes to show that collection happens way more often near city, especially within 1 or 2 hours travel time to cities. So what do all these patterns tell us? I think there are 3 important messages here. First, we see that, as we suspected, collection coverage is not complete in space, but environmentally it is doing better. Also, we see that collection is not plateauing in terms of species discovery, even in places where there are a lot of data already. And data also growing rapidly, particularly because of strong contribution from citizen science. I think these are really optimistic messages, indicating that we already know quite a bit about the data, or we know quite a bit about Australian fungi, but with increased collection in the future, we may find out even more. And where does this lead us? So this is just a small teaser for my future chapters in my PhD, where I will be taking the clean data set and actually trying to run biogeographic analysis to generate some knowledge about Australian fungi. Here, what we're looking at is a test product of biogeographic regionalization, where we use a simple clustering method and try to divide Australian landscape into different bioregions meaningful for fungi. So we're seeing that already with the data set we have, we can uncover some pretty realistic looking patterns here. So in summary, I have 2 main take-home messages. First, I think open access data already provide a really great opportunity to study the understudied kingdom of fungi in Australia. And I encourage people with research interest in this area to really take the opportunity and use the data, because it's really great that we have this resource here. And secondly, we see that if we increase collection effort, we can really benefit and know more about this fungi, this precious fungal kingdom that we know so little about in Australia. So any more data are truly welcome. And I would like to give a particular shout out to iNaturalist, which is a really beneficial and easy way to collect fungal information, and is very accessible for everyone. And to FungiMap, which is Australian homegrown citizen science organization and an effort to really increase our knowledge of fungi, which has really helped us to collect a lot more data in the last 2 decades. So coming back to the question I posed at the start, can open access biodiversity data help us? I think the answer is a cautious yes. There are still many barriers so far in terms of coverage and other issues, but I think the future is looking really optimistic, especially if more data comes in. So with that, I would like to give my thanks to my supervisors and colleagues, and to ALA, GBIF, and the other databases and repositories providing this wonderful data. and most of all, to all the people out there collecting fungal data who have really contributed immensely to the data that we can use today to answer all the different biogeographic questions. Thank you.
That's wonderful. Thank you, August. That's really interesting. I recently had a look at what the data holdings we have in ALA and realized there's 63 million animals and only 2 million fungi. It did seem a bit inequitable. So it's nice to know that someone's doing some really exciting work with that. But to move straight along before we come to questions, yeah, over to you, Jane. Love to hear what you've got to say. Thank you.
Great. So you can hear me and see my slides? Great. Okay, so thanks, Martin, very much for inviting me to speak, and congratulations again to ALA on this 10-year anniversary. First, I'd like to acknowledge traditional owners of the land on which I'm living and working. I'm in the Yarra Ranges of Victoria, and for at least 35,000 years, the Wurundjeri people have been the traditional custodians of this land. And I do pay my respects to their elders, past and present. Now, I'm a modeller, so I work on species distribution models, and today I'm just going to briefly showcase a few pieces of work that focus on fitting models with data like that from ALA. Now, species distribution models are models that can help us to answer some of the fundamental questions of ecology. So things like, why does a species occur where it does, or where could it occur? And most often these are a type of statistical model, and we're relying on the idea that environment largely affects where a species occurs. So we combine observations of species data here with estimates of the environment of the locations where the species has been seen, and we fit it in a model. And then that model can be used to predict to new locations as long as we know the environment of those locations. And with rasters or maps of environmental data like those in ALA, we can make continuous maps of predictions. Now, I'm calling these species distribution models SDMs, but they're also called ecological niche models and habitat models. And we can fit these models with various sorts of species data. So the records in ALA, and you'll see here currently there's about 91 million records, are presented as presence-only data. And presence-only data are data where we've got a record of the species presence, so a record of where someone has found the species, but no records of locations that have been searched without finding the species. Now, in fact, some ALA records might have come from comprehensive surveys where absences have also been recorded, but in general, modelers use all occurrences in ALA as presence-only data. Okay, so here are the current records in ALA for the yellow-footed antechinus in Australia. And the big question for modelers is whether these are a random sample of the species distribution, so indicating patterns arising from the ecological processes that determine that distribution, or whether they have imprints of biases in collection effort. And we think about this as the observation process, so the process that's led to the records. And as August has just shown you, biases are very common in presence-only data. Now, the problem for modelers is that these are very difficult to deal with in models. And I'll just quickly demonstrate the problem. So here we've got a toy landscape and we've got a species. These are the individuals of the species. And we've got a town down here on the bottom right. Now, presence-only data are the records where the species has been observed, so the ones with yellow highlights. But there's no information here about which areas were searched. Now, in contrast, survey data or presence-absence data show what was sampled. These red squares here, or quadrats. So here you can see that most of the sampling is close to the town, and in the 20 quadrats that are close to the town, only 4 are occupied. But up the hill, out of the 3 quadrats that have been searched, 2 contain the species. Now, presence-only data in collections like ALA are often biased towards areas that are easy to search, and it's understandable because it's easier to get there. Now, in this toy example, we've got more records close to town. And often environmental gradients are correlated with bias. So here it's warmer at sea level, close to the town. Okay, so we fit our model to environmental data, because the species is really impacted by environment. It's not impacted by the distance to the town. And so a model fitted to these data that ignores the sampling bias is going to see more records in warmer places. So it's going to predict that the species is more likely to occur in warm places. Now, survey data are much more informative. So they show here correctly that the species occurs less often at warmer temperatures. So you can have bias in the survey placement. We've got more quadrats close to town. But because you know this ratio of presences to absences, your model's going to be correct, even though it's more uncertain where we don't have as much data. So given that this one here is a correct relationship, we don't want to fit this opposite trend at the top here. And the issue is that we don't have the correct information within presence-only data to overcome the biases. Now, one question that's very sensible to ask is how much difference this actually makes in practice. And I'd like to give you one example. So this is Angela Muscatello, who did her honours project with me and Hani Huyala. And Angela's done a great job and just had her research published in Conservation Biology Journal. So Angela's study was based on data from the Greater Hunter region of New South Wales. She modelled 25 species, and these are the occurrence records for all those species. So there was 4 mammals, 7 birds, 11 plants, and 3 reptiles. And the species data were from both ALA and BioNet. And Angela's main question was whether the common choices that we make in how to model species distributions would have noticeable impacts on estimates of the best sites for conservation. Now, she used statistical modelling software Maxent in the free statistical software R for modelling the species distributions, and she used Zonation for making spatial conservation prioritisations. And she tested several variants of species modelling, so things like more or less complex models, different predictor variables, thresholding predictions, but also different bias treatments. And I'm going to focus on the bias now, although all of the results are really interesting, I'd say. For bias, she either didn't treat the bias or she used methods that measure patterns in observation effort across all species and use that to offset the bias. So here's an example for the anti-kindness that we saw previously. So on the left is her prediction that's using one of the better methods for dealing with bias. So this is called target group background, and it uses observations of all mammals to show where people have searched. And so notice where the highest predictions are, these ones in green. When we don't treat the bias at all, so on the right here, you can see that the predictions change substantially. They're a bit stronger along the coast, and we no longer predict these patches out in the west and the north. Okay, so Angela made predictions for all 25 species, and then she put them into a conservation planning software. And what that does is it ranks the grid cells in the landscape according to their biodiversity values. And this is 2 different prioritisations, and they're based on the species distribution models that treat bias in different ways. So have a look at the scale at the left here. The brighter colours are higher priority sites. Now, these 2 prioritisations are quite different. So, for example, on the right here, there's a great big patch down in the south that looks very important for conservation when we don't deal with bias, but when we deal with bias, its importance suddenly is degraded. And you'll see other differences too. So, for example, in the west out here. So handling bias in these species distribution models really is a big deal. And we do see this effect over and over again. Predictions change substantially depending on whether and how you deal with biases. But a difficulty that we've got is that all of the bias treatments using presence-only data have some problems. So what's the solution? So, well, one solution is to use data that helps us to pin down what those biases are. And this is just one example of some work I was involved in. The lead author here is Will Fithian, who was a PhD student with the statistician Trevor Hastie at Stanford Uni. And this is David Keith, who's an ecologist from the University of New South Wales. He was our expert. ecologists. So our main idea here was to combine the strengths of two types of data. So the collection data, like from ALA, is valuable because it usually provides a lot of data and that can help us to define ecological relationships. But survey data, so presence-absence data, help us to find the unbiased relationships, like down in the toy example I gave you. And so that helps us to deal with the biases. So it's addressing these ideas of ecological processes and observation processes, and it's trying to get both of them right. So in overview for the model lovers, Will developed an inhomogeneous point process model, and so that's like a regression model, and it estimates ecological relationships at the same time as estimating bias in the record. It's what we call a multi-species model. So we can make use of information from many species to understand biases. And we've got predictors for both environment as well as predictors for bias. And the important thing about this model is that we're combining presence-only data with presence-absence data in the one model. And we're not pretending that the presence-absence of survey data are presence-only data. We're actually using them as presence-absence data, and that's helping to strengthen our model. So we tested this on a large data set from these more easterly bioregions of New South Wales. We modelled 36 eucalypt species. We collated a set of predictor variables that were relevant for both the species distribution and also for bias. And so Will fitted all the models. And the new method worked very well. Now, the detail isn't important here, but this is just showing our performance statistics for each species down here, measured on held out blocks of survey data. And across the top, we've got different ways of fitting the models. And this one is our model with all the species and two different types of data. Now, higher is better here, and the blue lines show the best model. So you can see that the models combining all the species together with the two different data sets is tending to perform the best. And the models show that there were clear spatial patterns in bias in the presence-only data, and here they are mapped. So there's most collection effort near the coast in these data. So the main message here is that biases in collection data are common. They can be challenging to deal with, but the better bias correction methods are emerging. And these methods that integrate data from several sources are, to my mind, the way ahead. So all sorts of data could be combined. And the main idea is we're trying to get as much information as we can on the observation process. And if you're interested in looking at this further, this is a very good review paper that's come out this year that's looking more at this idea of data integration. So the models are still new and they're still being developed, but they are helping us to make the best use of the sorts of data that are in ALA. And they provide a very strong motivation too to collect data with more information about the observation process. Okay, so my examples so far have been species distribution models just for one snapshot in time, but often the real desire is to monitor trends in species over time. And then the issues with biases and issues with observation effort are even more important because observation effort often varies over time. So just to wrap up, this is another really helpful recent paper focusing on data collection and monitoring. In fact, some of you might know some of the authors. Steve Kelling is very involved in eBird in USA. And this paper again emphasises the difference between the ecological processes, so the drivers of species distribution, and the observation process. And it offers some really practical advice for people designing citizen science collection efforts to make the data as valuable as possible. And I really like this quote of theirs. I'll just give you a minute to read this. Okay, and because where we want to get to in the end is reliable predictions. These are predictions from eBird. They're showing the predicted migrations of this magnolia warbler from the equator here up to northern Canada. The paler tones are showing higher abundances, and you can see that time is ticking by at the top here. And this model and others like it are possible because of the quality of the data that's being collected, and because eBird have invested in computer scientists and statisticians who've helped develop models that suit these sorts of data. And some of their models are so well supported now by data and by evaluation that they're feeding into really important conservation decisions about critical habitat. And I think it's a really nice example of exceptional use of spatial data for biodiversity modelling, because it's one of the things I look forward to. And so finally, I'm part of the Quantitative and Applied Ecology Group and the Centre of Excellence for Biosecurity Risk Analysis at Melbourne Uni. And I want to acknowledge my students and colleagues there who've contributed to this work. So thank you.
Lovely, thanks, Jane. No, so much covered there. That's really fascinating stuff. And in fact, across all three speakers, thank you so much for giving your insights. It's really wonderful to see. So we've got another 10 minutes or so for questions, and we've got some coming on the screen, which I know none of you can see, so I'm going to filter them for you. So look, I guess my first question is that all of you talked about this need to detect trends. You talked about strengths and weaknesses in data. You talked about bias. So do you have a feel for what would be the one recommendation you would make if it were possible in an ideal world? What would you do that would make your jobs easier to do this sort of work? And Jane, you were speaking. Maybe we should start with you.
Okay, my one desire of mine is that if you're planning a citizen science survey, you do something to collect information on how the observations were collected. You know, you get some records of how long people searched, where they went. So and that paper by Steve Killing's got some really practical examples. So I just want more data on how the data have been collected. I want to know how they've been collected. Ideally, I want absences, but people don't always want to record those.
Fair enough.
But absence data would be wonderful.
Sure. I mean, Melodie, you talked a lot about low power to detect change in populations. Do you have an opinion on this matter as well?
Yeah, absolutely. I think that international collaborative efforts are absolutely key here, because the size of the task is completely enormous, and there are two parts to the task that are really important. One is collecting new data. We can only afford to rely on historical data for so long, and really it's only so good. We need ongoing data actual collection, and that can happen via sort of biodiversity observation networks. The Atlas is a wonderful example of that. That's actually shared its infrastructure globally, for example. GeoBon is an initiative that's promoting biodiversity observation networks supported by similar sorts of infrastructures and approaches and survey methods and so on. And then the other side of the issue is the advances we need in biodiversity informatics to support all of this fair data, data standards. And it's an enormous task, and we don't want to duplicate efforts. And so I think big international research collaborations in collaboration with NGOs and whoever else can support the effort is a really important thing.
Lovely. August, you talked specifically about fungi. I've seen some work recently about sequencing genetics of fungi from soil samples and things. So in some ways that could be an advantage. Do you have a view on where the future's going for detecting your target species?
Oh, yes, absolutely. So the soil sequencing part is a very exciting frontier for fungal research because, as Jane has demonstrated the importance of presence-absence data, it is really, it's a paramount shift for us to be able to get any sort of information of species absences. And soil sequencing provides that, or at least we are very optimistic that it can provide that sort of information. So it really complements the strength of the existing observational data we have. But at the same time, that doesn't mean that the existing citizen science data have become less useful. In fact, they sort of tackle two different aspects. So as we've seen in my talk, that coverage is really a strong concern of mine for fungi in general, that we need to know more about what's going on out there in places there haven't been a lot of data collection. Mm-hmm. And that frontier is still being moved on primarily by observational data. So I would say that the future will possibly move in a direction that hopefully we'll be able to integrate and combine the strength of soil sequencing data and citizen science observations.
Yeah, I can see how using the sort of methods that Jane was talking about could be a really interesting way of integrating those sorts of data. So one of the questions that's coming through on the channel here is that there's been a lot of questions. A few of you talked about time series, for example, and talking about trends. Someone was asking about environmental history, and so places with similar environment but a different past. And I guess a clear example of that is the recent bushfires and people's interest in those. Does that add challenges? Is that within our scope of prediction about different histories and their role? Anyone can answer that if you like. I don't mind.
Well, in species distribution models, you can include patterns like that if we know about them, if they're measured. And so if we've got good fire history, for instance, we can include in the model predictors for fire, and so we can get a feeling of how fire has impacted distributions. But often our problem is that we don't have the data, you know. Mm. And I guess there's fancier ways of including information that might be expert information, but yep, it's always tricky. All those things we don't know about.
Yeah, absolutely.
And it's a really good point. I mean, we've got a fabulous history of all sorts of things in Australia impacting our biota. It's very very lovely.
If anyone else wants to respond to that, we do have some time.
I just want to add that I think so the idea of environmental history is also linked in parts with data quality issues, because often what a difficulty we have in being able to model environmental conditions in the past in terms of with the species data we have is that the temporal resolution of some of the data we have are imperfect. So sometimes, especially with older data, we have a rough idea of that is recorded at which year they were recorded, but more specific information are sometimes not available. And also going beyond species data, it is also sometimes difficult to find environmental data that perfectly map conditions in the past. So it's adding on to that problem of, I guess, in a way, you want to be able to perfectly capture that perfect snapshot of what everything was like back in the days. And that will give us the most accurate information to track, I guess, long-term changes and trends. So really, I think the direction I would like to see in the future is for data repositories to expand the capacity to document more meta information and metadata about each individual record, so that we could have that information available for us if we need to model these nuanced details.
Lovely. Thanks for that, August. Look, I've had one question that's just really caught my eye. Melody, you talked a lot about the Iucn and the fact that our focus on rare species is missing a lot of particularly declines in common species. And someone's picked that up and asked the question, what would your recommendations be to the Iucn? Now, that feels like a big question, but if you want to tackle it, it's sitting there for you.
So the Iucn works with experts and works with existing data. So I think it's the Iucn could certainly, it's not my job to tell the Iucn what they need to be doing, but I think it's a joint responsibility for all of us to focus, in addition to rare species, on common species, and to look at monitoring common species, to raise the profile of common species. And, you know, they are being raised, their profile is certainly being raised by the sort of multi-species functional ecology approaches that are being done. But that will always be, you know, a species per capita function is the impact of that species on the environment is realized via its abundance and distribution. So abundance and distribution are absolutely essential biodiversity variables. And all of us as researchers, as citizen scientists, and the Iucn and other NGOs need to perhaps just start to profile the value of data on common species more strongly.
Lovely. Okay, well, look, there are still a few more questions, but we are sadly running out of time. We're coming up on the hour. So we may actually have to leave it there. But look, thank you so much for joining us and for giving to the audience for coming along and hearing these talks. But thank you so much to our speakers for sharing your insights. It's been really fascinating. If anyone who's listening would like to answer our exit polls on the way out, we'd be really interested in your insights. In the meantime, join me in thanking our speakers and have a good day. Goodbye.